This is lesson 2 of week 14. It is Life Sciences, Cell School, and of course, I'm talking to grade 10s. Grade 10s today we're focusing on cloning. Now, cloning is when a group of organisms are produced and they are all identical to the original parent organism. And of course, remember, we're focusing on tissues. So we are going to have what we call parent tissue. We're going to isolate one cell from the parent tissue, and from the parent cell, we're going to be able to produce 3 batches or 4 batches or however many batches. The number isn't important, but we're going to produce groups of identical cells. Cells that are identical not only to each other, but that are identical back to the parent tissue. This is what a clone is: identical to each other and identical to the parent tissue. Now, we may think that cloning is good material for science fiction novels, but in fact, this is how most unicellular organisms reproduce. Unicellular organisms such as bacteria, for example, when they reproduce, all that happens is their DNA duplicates and a wall starts growing and finally separates the 2 bacteria. Other unicellular organisms, such as amoeba, once again, their DNA would replicate and the organism would start pinching off and the cell membrane divides and we have 2 little organisms which can go their separate ways. But the important thing is that each organism is identical to the other organism and it's identical back to the parent. This is known as binary fission. Fission means to split and binary means 2. So one organism is splitting into 2 organisms, and these 2 organisms are in fact clones. They are identical to each other and identical to the parent, and this happens naturally. Certain plants can be replicated or reproduced asexually. Certain forms of lilies and other organisms that produce an underground corm or rhizome or a bulb, this can be split. You can dig it up, you can split the corm into 2, and each part can be planted and can grow a new plant. And if you tested the DNA, you would find that these 2 plants are identical. In other words, they are clones. Now, tissue culture is another way of producing clones. It is very similar to what I showed you originally, that you have parent cells or parent tissue, and one of the cells is isolated. It is allowed to undergo mitosis, which you have learnt is cell division, and once a group of cells is formed, you can further split that group of cells into smaller cell groups And each of these is a clone because they're identical to each other and they're identical back to the original parent. It seems rather simple to be able to talk about cloning in terms of unicellular organisms and even plants. But can animals be cloned? Well, Dolly the sheep was the very first mammal to be cloned. Scientists had had a great deal of success cloning lower animals such as, or more primitive animals such as amphibians, but Dolly the sheep was the first mammal to be cloned. In South Africa, a cow called Futi was cloned from the cell of an ear cell of a 9-year-old cow. So the ear came from an adult cow. Cells were taken out of the ear and they went into tissue culture, and after a series of extremely complex procedures, Futi was produced. And this all happened on the 19th of April. Futi was born on the 19th of April 2003. Of course, we can see that great ethical questions can be raised concerning the production or the making of clones and human clones. And these are questions that you have to think about in terms of the topic of cloning. Here is your homework question. What are some agricultural advantages to cloning? Well, we know precisely what the offspring will be like. So we have knowledge, precise knowledge of what the offspring will be like. And we know this because the clone is identical to the parent. And this is important, that the farmers may want certain plants to be identical to an excellent breed or strain of plants that they have originally produced. And if these offspring are cloned, we know that they're going to have all the qualities of the parent. In terms of cloning, we do not have to wait for the organism to go from seed into a little seedling and then go through the whole maturation period until it produces fruit or seeds itself. Tissue culture and cloning can help us to produce food a lot more rapidly. We can choose plants that will produce what people need. For example, trees that have good quality wood. And because we have knowledge of what the parent's characteristic was, we know that if we clone it, the offspring will be identical. That brings us to the end of our exploration of cloning. Tomorrow we will pick up on cloning as we look at stem cell research. For today, goodbye.